Okay, y'all, we're gonna get started with our seminar. There'll be a few stragglers coming in, so we'll sort of go slow. I want to introduce to you one of our own, Joel Fodrie. He has an interesting North Carolina history, and so I'd like to say a few words about that. He's the son of Eastern North Carolina. He came to UNC as a James M. Johnston Fellow, 95 through 99. Some very nice fellowship, very prestigious, hard to get, he got it. His undergraduate bachelor's degrees were in biology with highest honors based on an honors thesis he did, and history with a minor in chemistry. So he was all over this campus. His undergraduate research project won the R.E. Coker, Robert E. Coker Award, much tied to us, of course. And it was for work on the basin-scale population and our local sounds with the Peterson group. So that won the R.E. Coker Award. Most important, he almost made the varsity basketball team. I think last one up. Okay, so the day comes and you're going to go out and try out officially in front of with the boys.
So I tried out, and the next day Dean Smith retired.
He was so disappointed. But he did
decline in basketball.
Exactly, there was not going to be Joel. What happened was Joel scored 28 points, so the guy that was guarding him would be surprised. And but the guy he was guarding was Antoine Jameson, who scored 48 points on Joel. At any rate, almost made it. But instead, he chose to graduate with highest honors of all the rest. Part two in his career is he decided that it would be wise to get out of town because he wanted to come back to this neck of the woods later. So he went off to Scripps for graduate school to work with Lisa Levin, who used to be over at NC State some decades ago, a real terrific ecologist, biogeochemistry type. And there he finished his PhD work. What was it that Tom Wolfe said? You can't go home again? But here he is. So he broke that stereotype. After his PhD work, and I'll say a couple of things about the pups from that, he received multiple postdoc offers and chose one at Dauphin Island Sea Lab, where he worked with Heck. Was it Heck? Yeah. So his postdoc brought him also a research assistant professorship position there, and then we went to get him back and succeeded. I want to say a few things about his research contributions because they're all big picture contributions and very interesting. I'll just mention 5 of them. The scallop fisheries one we just heard about. Habitat influence on halibut populations, juvenile halibut, either where they are along the California coast. Very interesting paper on that, was part of his PhD work. Influence on climate change on Gulf fish populations that are frequent seagrass meadows. Another interesting paper. I guess the long-term answers are heading for the poles. They've got Florida in the way, but eventually they'll find their way around, I guess. After the Macondo blowout, a statistical look at what is happening to fisheries populations with some very interesting conundrums there. So
they're going to hear about it.
They're going to hear about that. Okay, I won't say any more about that. And now oyster reef restoration efforts and what we can use, how we can use food web dynamic ideas to deal with restoration processes. So Joel's a real Renaissance man. We're lucky to have him back as a colleague. I knew him when he was just a little undergraduate in my 101 class, now 401 class. So Joel, welcome back.
So I'm one of the most long-winded people alive. So I'll try to be quick. This is a true story, and I was telling somebody earlier. When I was looking at grad schools, I was basically down to Duke and Davis and Scripps. And I was in a hallway, and you may not remember this, about past Chris Martin's.
Very dangerous.
And he asked, well, what are you thinking? How's it going? I said, well, I'm probably leaning towards Duke because I like the guy there and it's good research. He's like, no. You got to go out west, get away from here. And this is a true story. Like I went into that hallway thinking I was going to go to Duke, and I left that hallway having decided I was going to go to Scripps because of him. And so this is his fault. You're in here today. All right. Well, it's great to be back. Kind of surreal. Our lab, I hope, does a lot of neat stuff here in North Carolina at the field site. Unfortunately, I guess I'm not going to talk about that today. If I don't screw it up, maybe I can come back sometime in the future and talk about that stuff, which I think is cool too. But I know a lot of you work in the Gulf, and I thought I'd touch on some of the things that I've been doing there, some of the connections we might have. The other thing this talk is not is this is not really a talk about some central concept. You know, a lot of times the talk will be focused on predation or diversity, and this talk is not that. This talk is really just about, I've been working in the Gulf for a little over 10 years. You know, that's a decade. And this is how I see the Gulf shaping up with all the different drivers that may be forcing it. And it's really just about the detective work that we've been doing to try to do like emergency room science, right? So there's all these things that are affecting the Gulf, and we're trying to keep up with how is this changing? Like, is it good or bad or indifferent? So with that sort of meager introduction, I will say that if I was going to try to wrap this talk around any big picture concepts, I'll come back to this idea of press versus pulse disturbances, right? So you can have some system that's motoring along in some equilibrium or whatever, and you perturb it somehow, you disturb it. And generally speaking, we can think of those disturbances as being like presses, where the disturbance is now like constant, like you're constantly forcing the system with this new dynamic. Or you can hit the system with some sort of pulse. You might think of a hurricane or something, like pulsing through it. And so a lot of the things I want to talk about can be categorized as either press or pulse, and I'll try to make some broad brushstroke conclusions about what I think may be important in the Gulf. More specifically, I'm just going to talk about three things that I've been looking at in the Gulf: warming, oiling, and then fishing. I did give a talk here several years ago and kind of did some of this fishing stuff, but hopefully some of y'all have forgotten and some of y'all haven't heard it. So I thought I'd revisit it, and I do think it's kind of fun. So I just want to talk about it. All right, so first up is I want to talk about warming. It's not surprising. We know places are getting warmer. The Northern Gulf is no different. The graph here is sort of 30 years, and this is the average minimum daily temperature during the summer and fall. And it's kind of a shocking amount of warming. It's like 3 degrees in terms of the daily minimum temperature that organisms are experiencing in this part of the world. And it's the minimum that we care about, because that might be what really keeps things out of a new area as they're trying to move in response to climate. Like, it's not the average temperature, it's the max or the min. And so, you know, we know that other places are seeing responses to climate change, like changes in primary production or ocean acidification. Sea level rise is a big deal. Really interested, I won't talk about it much today, but changes in the phenology or the seasonal timing of things, increased storminess, and then largely what I want to talk about are these poleward shifts. And the Gulf is a pretty interesting place to think about it, because there's actually a limit to that. And I don't think we're there yet to understand what's going to happen, but this is not a place where things can just keep migrating north. They're going to run into a cap, essentially. And so I think this is a place to be worried about polar ring shifts. Nothing that I do really in this talk is rocket science. It's far from. It's not very complex. The difficulty is just in trying to get numbers to understand fish populations. Fish populations are really noisy. They're patchy. And so the challenge is sometimes just to do the amount of work you have to do to get the numbers to basically shrink your confidence interval down to a point where you feel comfortable saying something about fish populations. So I showed up in the Gulf in 2006 to work on a project about nursery habitats. A lot of marine critters have nursery areas. That's not so much a terrestrial concept, but in the marine realm, it's a pretty big deal. And we were trying to understand what areas of the Northern Gulf, from the Chandelier Islands in Louisiana all the way over to the Florida Panhandle, were the important nurseries. And for that project, I was tasked with going out and collecting fish that I was then going to do further work on. But as we were out sampling, pulling a net basically through the seagrass meadows, catching things like these gray snapper, I kept bringing things back to the lab, and Ken Heck, my mentor, a guy named Rich Aronson, really knowledgeable ecologist, they kept saying, well, what's that doing here? That shouldn't be here. Like, we haven't seen that before. And at some point, they just kind of egged me on to try to find some way to do some comparison of what used to be and what is now, and has there been a change? And as it happened, Skip Livingston had also worked in the Northern Gulf. He had used basically the same gear. Ken Heck, my mentor, was his student at one time, had been working on those sampling programs with Skip Livingston. And so we knew that we could probably make a comparison between what he saw in the 70s and what we were seeing in the 2000s. And so again, we both used essentially the same gear, all the same methodologies. His sampling was more spatially limited and temporally expansive, and ours was more spatially expansive, temporally limited. But we both caught about 125,000 fishes. And so we thought we could do this comparison of what he caught and what he caught and what we caught. And so just very sort of pictorially, this is the fauna that Skip Livingston sampled. And if you know our fishes here locally in North Carolina, you might recognize some of these beasts. Here's a pinfish. Here's a spot. There's a pigfish, silver perch, filefish, pipefish. Essentially like a very temperate fauna, okay? Like you'd recognize this in North Carolina. And in reality, we see all these fishes in the Northern Gulf still today, with very few exceptions. These guys are still here, they're still players. But in addition to those fishes, we were catching all of these guys. So it's multiple serranids or your groupers, it's multiple snappers, it's parrotfishes, surgeon fishes, butterfly fishes, you know, things that you just automatically know because you've been around, have a distinctly like tropical vibe to them. And in fact, when Livingston was doing his sampling, these more tropical species were 1 out of every 600 fishes. Every 600 fish you catch, one of them will be a tropical. When we did our sampling, 1 out of every 20 fishes was one of these tropicals, right? And so at some point you might start to think, well, this is kind of a notable change in the dynamics of what's going on. And there's 4 more species that we think are relevant for looking at these range shifts, shown here. So this guy is a lane snapper. Skip Livingston never caught one in 10 years. And when we sampled, it was the 7th most abundant species that we caught. Gag grouper and gray snapper, Livingston caught them. We caught them at like 2 orders of magnitude greater rates than what he saw. And then this guy right here, this emerald parrotfish, which I'll come back to, we caught it about a 25-fold greater rate than what he sampled. Okay, and so, I mean, there are some players here that are really common now that weren't in the past. And it's not just fishes. You know, there are these sort of distinct climate-related fingerprints that you can pick out of different people's papers. So in the Northern Gulf, they're starting to see aquapora corals. While I was in my postdoc, the first reported catch of like Goliath grouper or red grouper in Mississippi happened. The first reported catch of a bonefish in Alabama happened. There's increased sightings of manatees. Now, whether or not that's related to there being more manatees or there being more people is debatable. And they're also worried about things like this tropical ciguatera poisoning. It's actually showing up now in the Northern Gulf, right? And so all these things are kind of happening at the same time. And just to bear that point out a little bit further, this is a picture taken from the northern tip of the Chandeleur Islands. So the Chandeleur Islands are there on that eastern end of Louisiana, just south of kind of like Mississippi, Alabama, Maine. And what you're looking at here is you're looking at, you know, the seed of a black mangrove, and you're looking at black mangroves that are now growing right in the marsh. So in 2006, the first year I went, there were just like 1 or 2. And it was like, what are those things? We went over and kind of confirmed that they had the nematophore sticking up and they were black mangroves. And basically now, if you go there, it's just contiguous mangroves kind of right there along the marsh. And mangroves have actually showed up on the islands like in Mississippi and in Alabama, right? So it's kind of happening at timescales you can even see if you're out there enough. And to go along with this mangrove picture, I just want to revisit gray snapper. This was a separate report just looking at the recreational harvest of gray snapper in Louisiana. And over about the last 20 years, the increase in harvest rate has gone up about 2 orders of magnitude, which is the exact same sort of increase that we've seen in terms of juvenile abundance. And the reason that I showed this graph here right after I showed the picture of the mangroves is gray snapper have another name some of you may know. They're called mangrove snapper. And so it's probably not surprising that these things would be kind of moving up together. So I think there's, you know, a real signal here. The other point that I would like to make, which may be a bit controversial, is that in terms of fisheries, I think you have to view climate change as there's going to be winners and losers. If you like catching gray snapper, this is not the worst thing that's ever happened to you in Louisiana. And that's not specific to that part of the world. Here in North Carolina, in my lifetime, we're seeing a lot more stone crabs. You know, the reported northern range limit of stone crabs used to be Cape Lookout. And now they're super abundant around Cape Lookout and probably go maybe as far as Cape Hatteras or something. And stone crabs are delicious. And so if you like stone crabs, you know, maybe good. But stone crabs are also eating other shellfish, clams and oysters. And we don't yet understand, again, who all the winners and losers are going to be, perhaps. But there probably is some of that dynamic. So this is also just another representation of this shifting in the fauna. This is the northern range limit of species, just pulled from fish base. These are species that we lost. So by that I mean Livingston sampled them, but we didn't. And then there's species that we gained, meaning that Livingston never caught them, but we did. And basically the species that we lost have a farther northern range limit than the ones that we gained, right? So I've done my best to try to convince you that it's happening. We also wanted to go a little further and ask, well, what are the implications of this? Does it matter? Like, is there just niche packing and everybody's playing along happily? Or are there going to be consequences of this bringing together of temperate and tropical fauna? And so to bear this out a little bit, pinfish are in the Gulf, they're 50% of all the individual fish you catch in these estuaries. I mean, pinfish are super abundant. We had noticed there were a few places, though, that strangely enough, you didn't catch any pinfish. So a place like Horn Island, you might pull the net, and what you do catch are a ton of lane snapper and no pinfish. Places like the Mobile Bay, you catch plenty of speckled trout and then lots of gray snapper and no pinfish. So is it possible that somehow these snappers have come in and perhaps excluded through some mechanism pinfish? This was sort of really hard to test. What we came up with is some sort of Bakke design. And Bakke means before, after control impact. It's just a way to try to look at impacts of some event, an oil spill, an invasion, climate shift. The idea was we picked these 6 sites. At half the sites, in the summertime, you get pinfish. They arrive first. And then snapper don't arrive ever, okay? And in this system, pinfish arrive first as juveniles, and then the snapper come in later. That's just their life history, their seasonality, chronology. The impacted sites were the sites where the pinfish showed up first, and then the snapper did show up after that. And so by looking at the interaction between time and this impact treatment, we were able to ask, do the pinfish here at these sites respond differently in a way that maybe they respond differently than the control sites in a way that may reflect what the snapper are doing to them. And this is, I think, the ugliest graph I'll show. Essentially, we wanted to look at 2 metrics of impact. One was, what did the arrival of snapper do to the abundance of fish? And that's shown here. And the abundance just comes from the catch rate. You know, you pull the net a certain distance, you get a catch rate, compare those. The story here is that abundance seemed unaffected by the arrival of snapper, okay? At the sites where snapper arrived, the pinfish didn't seem to show a response. We also wanted to look at the growth rate of pinfish. That's what's shown here. And the way we got growth rate is bony fishes carry around a little structure in their heads called an otolith. And like a tree, it grows in rings, and that's shown here. And you can look at the gaps, the distance between those rings, and these are daily rings. And you can get an idea of how fast that fish is growing or has grown before you capture it. And so we looked at the growth rates of pinfish at impact and control sites. And the story here is basically that the pinfish at the sites where the snapper arrived did seem to grow somewhat slower, but it wasn't like so obvious that you could confidently say that those snapper are retarding the growth of pinfish. It's in that gray area that we hate to be in, where there may be something, but it's not absolutely not dead clear. So that's sort of a sideways interaction, right? The competition between fishes that look the same and are about the same shape. We also were curious about potential top-down effects in these seagrass systems. So seagrasses temperate seagrasses and even these sort of subtropical seagrasses, we don't think there's a ton of herbivory that mows down the grass canopy. But there are some animals that we know do that, like manatees or green turtles especially. This grass here that you see here is called turtle grass because the turtles like to munch it. And again, the manatees and green turtles, they're probably maybe becoming more abundant in the northern Gulf, is what anecdotal data kind of suggests. And we wanted to look at this guy, the emerald parrotfish, because any parrotfish you catch, emerald parrotfish you catch, if you cut open its stomach, you get this quarter-sized dollop of munched up grass. I mean, they're just little grass-eating factories. And so we basically did some lab trials and some field surveys of what's in their guts from the wild. And parrotfish leave a very characteristic bite mark shown here. It's not hard to see when parrotfish are biting seagrass. And if you look at the amount of seagrass, the things like parrotfishes or pinfishes or filefishes, about the only other known herbivorous fishes that eat grass, the parrotfishes are way better seagrass eating factories than the other species. And we did some envelope calculations to look at what the impact of a higher density of emerald parrotfish might be in the Gulf. And so this is a photo stolen from Bermuda, where a group of researchers set up these turtle exclusion devices. And they wanted to know what the seagrass does in the absence of turtle herbivory. And what I hope you sort of see visually here is that in places where the turtles can't go eat the grass, you get this higher standing biomass of the grass. And in the places where the turtles can forage, they just mow it down to nothing. And so we were basically asking the same thing of what are parrotfish going to do now that they're becoming more abundant in the northern Gulf. And our envelope calculation is that the amount of parrotfish currently in the Gulf now, they're eating somewhere between 5 and 25% of the daily seagrass production. So if you used to have seagrass that was a meter tall, now it might be somewhere between 95 and 75 centimeters tall. And this may be important because the structural availability of seagrass is pretty well correlated to the amount of nursery role that it plays and the ability to produce shrimp and fishes that we like to catch and eat. And so we're worried about a world perhaps where there's more consumption of the grass and that may reduce its function for us as habitat. So there's some interesting things going on there. Now I want to leave that though and just talk about oiling. I got into oil spill research very accidentally. Had been surveying the Gulf starting in 2006. When the spill happened in 2010, we had before data, and it allowed us to get involved in, okay, what's happening with the oil spill. Lots of people at Chapel Hill are involved in this. UNC is very well represented. If you didn't happen to know, the oil spill happened about 100 kilometers offshore in 2010. It lasted like 3 months. Most of the northern Gulf experienced some degree of oiling. And we're worried about what that does to fisheries in particular, because we know from previous oil spills the pathways in which oil can impact fish. So just the physical gumminess of the oil can screw up the fairly fragile feeding and breathing apparatuses that fishes have. It can also reduce their ability to produce mucus, and without that mucus, they're more susceptible to infections. We know that the toxic pHs of weathered oil really affect fishes, even at low concentrations, like 1 ppb concentrations. You can see growth effects or genetic responses, reproductive cost. We know that from Exxon Valdez and other spills. And something that like Pete Peterson has been big in promoting since the Valdez is we need to worry about these chronic or sublethal effects. Oil in the sediments can keep having effects on fishes that lay benthic egg masses perhaps for decades. So of course there are reasons to be concerned about the effects of oil on fishes. I've got this clicker, I keep forgetting. However, big shock, this is a complex system, right? And so let's think about the ways that oil could impact these systems and the way that it can propagate or attenuate across different hierarchies. So we know, for instance, that oil can be bad for fish. It can gum them up. It can be toxic. But oil also affects the things that eat fish. oil affects the habitats that the fishes depend on. In this case, in 2010, oil did things like change the phytoplankton community that ultimately the fishes may be getting their energy from. Oiling turned off fishing. And we know that fishing has negative impacts on the abundance and size distribution of fishes. And so even if oil may impact individuals, it may not be clear how it affects populations or whole communities. And then on top of that, we've got all these other stressors like habitat disturbance and regional warming. And so how do we begin to pull apart and understand where these effects are evident and what it all means? So I want to talk about a couple of studies that aren't ours, where people have looked at how organisms respond. And so this was a study led by Andrew Whitehead, where they went out and they collected fundulus, Cacao in the Gulf of Mexico, and they collected it at different sites. And they did that essentially as soon as the spill began, before oil reached shore. Shown here, this is sort of pre-oiling. They collected at these sites. And then they collected during the oil spill, and then after the spill had at least been capped. And so this is a combination of sites that did not see oil. All of these basically never saw oil during their study. And then this one site right here that did see oil. And so this is that Bakke design again, before-after control impact. They've got before from all the sites, and they've got after from both the control sites and the impacted site here. And they wanted to look at a suite of responses, physiological, genetic. I'm just going to show one sort of pictorially of how fundulus responded to oiling. So this is the oiled site before, during, and after. This is a control site, and this is a thin section of gill tissue. And what you see here is this blackening, this gumminess. This is like smoker's lung or something, right? Where these fish are experiencing some sort of damage to their breathing apparatuses, not seen at the control sites. They report a range of genetic responses, physiological responses. These fishes are showing lots of impacts of the oiling. That study was in PNAS. Here's another one that's in PNAS. These researchers looking at offshore fishes like mahi-mahi, or I'm pretty sure this is a picture of amberjack. I have forgotten. But this is a larval amberjack that was grown in the lab, not exposed to oil. And this is what a healthy looking larval amberjack looks like. And then here's an amberjack larva that was exposed to oil. And the eye is poorly formed, the heart is poorly formed, the yolk is incompletely developed, the spinal cord isn't developed correctly. This isn't what I do, I'm no expert, but this is a jacked up looking fish, right? Like this fish is not happy, it's not healthy. And you can imagine that if all the fishes are subject to these sorts of responses at very, very low concentrations of oiling, that's bad news at a population level, right? You think that these types of responses would show up and you'd have less fishes. And so there's a couple different ways you could look at that. You can imagine a world where you've got data pre-oiling and then after oiling at a site, you know, how many fishes were there before and then after the oiling. You could go to a lot of sites and look at places that weren't oiled and then places that were oiled. In either scenario, what you expect to see is that, you know, either before the oil or at unoiled sites, you probably should see more fishes than you would at an oiled site after the spill, given how dramatic these organismal responses appear to be. And so no one goes out and really samples larvae effectively. They're too diffuse, they're too patchy, they're too variable in space and time. But those larvae ultimately become juveniles. And in the marine realm, most of the time those juveniles are going to concentrate themselves somewhere. Again, this nursery sort of role that habitats play. And so what has been looked at a lot are how many juveniles showed up in 2010 versus before, or at sites that were oiled or not. Because that juvenile stage has integrated all of those larval vital rates, right? And so if the larvae are impacted, you might expect the juvenile to show some numerical response. So I've been a part of a couple of those studies because I usually work at the population level. And so first, keep in mind, I already mentioned this, we were out in 2006, 2007, 2008, 2009, sampling all these seagrass meadows from Louisiana over to Florida. and we were sampling them pretty daggone well. And so what you're looking at here is you're looking at the catch rates of all fishes, pre-spill, and then the year after the spill. And this is Louisiana, this is Mississippi, this is basically Alabama, and this is over in Florida. And there's certainly no clear signal of catching fewer fishes in the year after the spill than the year before the spill. Now, this is largely a pinfish graph, because pinfish are most of what you catch out there. So I don't want to do some sort of flip-flop of talking about like Fungulus as a lab rat and then pinfish as a field organism. And I know you're not supposed to really show a table in a talk, but this is a pretty simple one. This is a range of species that we caught. So like pinfish, another cyanid, that's a hymulid, there's a snapper, another cyanid, that's a pipefish, another cyanid, toadfish, catfish, parrotfish, there's a serranid or a grouper. This is a wide range of species types. And all you need to basically look at is what was the trend from before spill to after spill? Was the trend that we caught more after the spill, we caught less after the spill, or we saw no change, statistically speaking? And of the 20 most abundant species that we surveyed, the only response that we saw was actually more fish after the spill. And I think this is 12 species showed that response. And this is like a statistically significant response, not just a trend, but like the error bars are such that we could say with some confidence that it looks like there was an upward shift. All right, but we're still not talking about Fungulus. Oh, sorry, I forgot this slide. So these are those same sort of data looked at slightly differently. Those were sort of univariate responses. This is a multivariate plot. So each dot here is essentially a trawl sample that we conducted in Louisiana, Mississippi, Alabama, and over in Florida. And these plots are multidimensional scaling plots, which basically take multivariate data sets, like your catch rate of species A, B, C, D, E, and it basically boils them down to two axes shown here. So this spot here represents the entire community that we caught in one sampling with the trawl. And the key difference is that the farther apart 2 points are in this space, the more distinct those catches are, right? And so if two dots fall right on top of each other, that basically means they look very similar in terms of the overall community structure and catch rates of the entire like fish assemblage. And so the colors here are basically pre-spill, and the black dots are after the spill. And the black dots aren't really any different than the before spill data. There's no like community shifts that we can obviously see. Now I do want to talk about Kakaho real quick, or the Fungulus, because we've also sampled those. So we worked over in Barataria Bay and Terrebonne Bay in Louisiana, going to lots of different sites where we were had data or knew that the sites either received oil or didn't receive oil. And so we sampled, I forget, 30 different sites, 15 oiled, 15 unoiled. And we looked at the catch rates of those marsh fishes, like Fungulus, which Whitehead et al. Had also looked at the organismal response. And so here are some of these marsh platform fishes, like Kakaho, or diamond killifish, sailfin molly, sheepshead minnow. And again, here are the catch rates of fishes, unoiled sites and oiled sites of 4 different species, 3 different times after the spill. And just to summarize it, it's very rare to go to a site and not catch as many of these species, each of these species, as you did before the spill. Maybe the most significant result we ever saw was at one time point for Kakaho, we actually saw more fishes at the oiled sites than the unoiled sites, okay? So to try to bring all this together, we published a paper a couple years ago, and at that time, there had been 24 studies that had looked at how organisms respond to oiling. Some from the field, some from the lab, different types of species, looking at reproductive responses, genetic responses, physiological responses. In 97% of those cases, they see some sort of impact. Like, just go ahead and write it down. Like, if you do the experiment, you're going to see an impact, right? And these are valid studies. I'm not critiquing the studies, okay? And it's not just lab studies. These are organisms taken from the field too, some of these studies. However, those of us who've been out looking at populations, it's only been 6 studies, but those 6 studies have been a number of designs, a number of places, covering over 120 species. And across all of those studies and all of those species that we've looked at, in 99% of the cases, you don't see any population level response. And that kind of sucks, right? Because like, how do we tell people what's going on when there seems to be this huge paradox in the literature, and that your impression of what happened sort of depends upon your method, obviously. Like, how do we resolve these like pretty obvious differences based on how you do your studies? And I've largely focused on the fishes, but there are other important nekton people have been looking at, crustacean-y things like blue crabs and shrimps. Typically, people think of the crustaceans as being more sensitive to oil than fishes. But in the studies that I found, 5 studies looking at organism responses, 80% of the time you see some impact. Studies that have looked at population level responses, 80% of the time you don't. So this difficulty in detecting an impact seems to be pervasive. Unfortunately, this is like a 15-minute slide. I've got like 1 minute to go through it. How do we start to reconcile these different responses that we seem to be seeing? We proposed there were 2 things that could be going on. Number 1 is that populations are responding, it actually is happening, but we can't see it for some reason. Like, something is just making it impossible to see the effect. The effect is happening and we can't see it. There's also this suite of mechanisms which could be buffering populations in a way so that even though you might see an organism respond, the population never would. And I won't go through them right now. I want to show you a couple examples and we'll come back to this. One of those factors might be what I mentioned before, and that was we turned off fishing in large parts of the Gulf in 2010. So these are data for spotted sea trout, or around here we call them speckled trout, shown here. So this is a trait of speckled trout, Louisiana, Mississippi, Alabama, Florida, the 4 years prior to the spill, and then 2010 after the spill. This is a log scale. And over in Louisiana, Mississippi, the catch rate of these little guys was like an order of magnitude higher, okay? Now, if you know speckled trout fishing, people are out there starting right about now. Every knucklehead like me that are trying to catch their 2 speckled trout. And these fish spawn in the summer. And so most years you're out there removing a lot of the mamas and the papas, and they never get to spawn. In 2010, that didn't happen. We come along in July, September, October with our sampling, and we're catching a lot of these little small fish that were spawned in that same year. And so again, we just can't understate what turning off fishing did to this system. That was like the real experiment, like the oil for these fishes. Like the oil spill was just like a way to do the actual experiment, which we don't have really good data on what actually happened. And there are other sort of complicating factors. This is a really ugly, but hopefully accurate marsh food web that a group of us, including Shelby Ziegler, who's in our lab, put together a couple years ago. And what we were trying to do was ultimately get to this graph over here, where we have 2 axes. We wanted to know how sensitive are different players, how sensitive are they to oiling, and then how important are they in the food web? Because like, if you're really sensitive to oiling, but you're not important to the food web, that's not where we should spend our time and energy understanding responses. You know, we wanted to understand what species are kind of up here, like who are both sensitive to oil and who are the key players in the system. And things that show up over here are things like gulls, birds essentially, and blue crabs. What do all those things eat? They eat little fishes. And so we might have these food web cascades that are offsetting the impacts that the oil was having on fishes. So typically, little fishes die at the mouths of a crab or a bird, and this year they died to oiling, but it's kind of the same number of overall deaths, perhaps. This is very speculative. I'm not burdened by lots of data on this one. You know, these are sort of spatially complex systems. This graph is kind of busy. The only point is, I'm interested in what nurseries are important for things like snapper and grouper, because we don't currently know. We don't know what areas are really seeding populations. For the little fishes that we're sampling here, we don't know where the larval supply is coming from. Is it local? Is it somewhere to the east? Is it somewhere to the southeast? And so spatial subsidies can like blur the lines between oiling impact in ways that we're probably just not going to understand. So we have these factors that could make it hard to see population level impacts, like fishing, which I think is a real thing, like these cascading food web effects, which I think are a real thing. Maybe worried about lagged effects. Maybe the effects are kind of building, the instabilities are building, and we're only going to see them now. Every year I sort of just get a little tense thinking about, is this going to be the year where Menhaden collapse or something? One thing that I don't think is a big part of the story is this concern about fish is just being noisy to begin with. People worry about fish populations are so noisy. How do you ever detect them statistically? I would argue that populations are noisy, but communities aren't usually, and we're not seeing community responses. And even though populations are noisy and we're worried about seeing a response, I would argue we are seeing a response. Like statistically, we have enough power to say there's been a response. It's just the response has been, there have been more fish after the oil spill than before. So I don't think that what would probably be my initial knee-jerk concern is actually a big player here. And then things that may actually just buffer populations. This has been much harder to get at, but it guides where our research might go. The big one is just fish populations are known to have these compensatory effects, essentially these density-dependent effects. If you reduce the density of a fish population, they already start to experience less growth penalties and mortality penalties. And so the oiling, early on reduced the density, but those fish that were left may have done better. These are compensatory processes that we know happen in fisheries already. Okay. So then the third part, fishing. If I had to sort of try to put this part into a bigger tent, I'd start with maybe fishing down food webs. So Daniel Pauly et al. In the 1990s made a big contribution talking about the trophic levels that we used to fish and the ones that we fish now. We used to fish the tuna, now we're fishing lobsters, in the future we may be fishing algae or whatnot. This sort of downward trend. But not only across species fishing down food webs, we're also worried about, we used to fish big tuna, now we're fishing little tuna, perhaps. And how do we recover those data? Because they're really hard to get. And the time scales are decades, and we often don't have those data sets. And there are hypotheses about like the role of big fish being disproportionately important. There's one, it's called BOFs, the big old fat females. They are really good baby making factories. And you'd rather have one big female than 10 smaller females sometimes, because that gives you more reproductive output. So we're really concerned about keeping big fish in the system. And big fish are important, big fish are awesome. This week, I need not say more about that dynamic. But how do we recover the loss of these big fish when it's happened over a century or 5 decades? Fishery scientists often lament that we don't have ice cores. We don't have that record to go back and look. So we're always looking for clever ways to do it. This is a data set published by Laura McClanahan a few years ago, where she went into old newspaper photos in South Florida, and she just looked at the size of the fish that were up at the dock when the boats came back and people took pictures of the catch. And what she saw in these photos is that over the last 50 years, the mean size of fishes in those photos had gone down dramatically. And I think this paper made a really big impact. And we would love to have more ways of recovering those sorts of stories to understand what's going on. So kind of by accident, I got involved in looking at these records and these different tournaments that happened in the Gulf of Mexico. So I was actually interested in an angle of that climate change story, and went and asked about what records these tournament has, and got an answer I didn't like, and so started trying to find ways to get more information, and that led me accidentally down this path. But it turns out that in Alabama, there's an 80-year-old tournament. In Mississippi, there's like a, what would that be, 70-year-old tournament almost. And there's one in Texas that's like an 80-year-old tournament. And if you go back and look at the records, you see things like, here's a big grouper. That's a 6-foot fence. That's an 8-foot ladder. This is a 400-pound grouper. And we'd like to know, like, is that the way the world used to be? And does it still happen today, or does it not happen anymore? And so the way that I ended up getting these records was going and looking at newspapers. And so I went to the library, I had a library card, and I could very reliably go look at the newspapers from all these different years, and they were publishing the leaderboards. And so if you woke up one morning in 1962, you find out Marilyn Monroe had died. But more importantly, for our purposes, this tournament was happening, and they were giving us the data. And so this is data for like 25 different species. And, you know, we've got this record every year, very reliably. These tournaments happen the same time every year. They basically have the same rules. And so it's almost standardized, right? And I'm going to go through this quickly just to get the main points, but we've sort of broken the species down into 4 groups. The highly migratory is like your tuna dolphin, the reef-associated, your snapper grouper, barracuda, amberjack, estuarine fishes, flounders, trout, sea trout. And then there's a group, the coastal-pelagics, which is based on how things are managed and their life histories. So it's things like bonito, sharks, bluefish, triple tail. It's kind of a catch-all, but it's a real, I think, ecologically sound group. And so what do these data tell us? It's not even just pictures like what Lauren had, McClanahan had, it's actually numbers. I don't want to cheat you out of a few photos, although I'm getting short on time. And going through these records, man, you learn a lot, and it's just fun. And so, for instance, here's a photo from the archives, and I like this photo because, again, it's a nice big tarpon. It has like a ghostly sort of sense to me. But I also want to remind myself is when you go through papers, if you ever have the chance, for a city like Mobile or Morehead City or Boston, understand that historically, the water was their lives, right? And so like in Mobile, every time a fish kill happened, and those fish kills were probably because of low oxygen, it shows up in the paper. And they call them jubilees. You know, every time a storm comes through and does something, it shows up in the papers. And so me not even expecting to see this stuff, you just start seeing all these stories and you can think about, well, is that a record I could use? And there's tons of biases you may want to avoid. But I found this stuff very enriching. And I like this photo. I like a lot of the photos because if you've ever read like any Steinbeck, like Cannery Row, like, man, it looks just like that, right? And this is like 1930s, Great Depression. Like, look how skinny those dudes' arms are. Like they weren't eating a lot back in the day because it was just hard times. And here's a photo of some people that I kept meeting in the papers. I don't know them, but I feel like I know them. And so this lady shows up like in the late 70s, and I actually can't ID that shark. And then here she is again in the early 80s. And in this case, it's pretty obviously a tiger shark. I can see the bars on the side. I like this photo for a couple reasons. Number one is we very, very rarely see sawfish. And in fact, in 1960-something, they caught a sawfish, and they said it's the first one we've ever caught. And that's not true because they actually caught one in 1931. The other thing I like is this is the way they dress to go fishing, right? And I always say when I get to this point, like when I started duck hunting, I went with the guy that wore the hat and the coat, patch, and the tie, like Elmer Fudd. And like they actually did dress that way for this type of sport back then. I think this photo got slid in. I don't think it's a tournament because that looks like some sort of commercial fishery, but like what a big stingray. I think you can see the stinger. It's like as long as my hand. And so, I mean, wow. And there was this thing. So when these tournaments started, like the two big species were tarpon and the devilfish, like the giant manta rays. And tarpon and the devilfish actually disappeared from the records within like the first 10 years of fishing because the fishermen basically said we've caught them all and we had to stop. And this is like conservation, you know, in the 30s. And so we don't even include these species, tarpon and devilfish, because the records aren't complete. All right. I just need to make the point, in full disclosure, these are not fishery independent data, like not the way that we do science. These are distinctly fishery dependent data, meaning there are biases. We try to embrace those biases because sometimes they actually work to our advantage. So for instance, over the course of these rodeos, there's been more and more participation. So all things being equal, you're sending out more and more people to sample. You might expect to see, well, the size of the fish isn't going to remain constant because now you're putting more effort into it. So maybe the size of the fish will be getting bigger and bigger and bigger just because of effort or because of bigger boats or GPS or better fishing tackle. So what is your null is kind of hard to define, all right? I'm going to skip over the highly migratories and the estuarine species, but I do want to show you data from one of those reef-associated species, red snapper. In the Gulf of Mexico, red snapper is king. Right now, Sea Grant in Alabama is putting out a $10 million RFP for one group to try to figure out how many red snapper there are in the Gulf of Mexico, right? So this is an important fishery. And I've kind of dropped the error bars and just want to show you sort of the general trends. But these are the sizes of red snapper that have been caught in the rodeo across 80 years. And in all three rodeos, the trend is basically, the black dots show pretty well. Wow, the fish are actually getting bigger. The white, the fish are getting bigger, maybe plateauing. The fish are actually getting bigger. And so that's good. That's a sign there's at least some big fish out there. We're not prepared to say how many, but there's some. You can go find them. And you might be saying, okay, but again, he just said this could just be an artifact of better gears, more people participating, that this is not like a real ecological signal. Well, it turns out that for red snapper, the tournaments were allowing scientists to pull those otoliths that I mentioned before out. And so what we have is we have a species that otoliths have been pulled, we can analyze them. One of those fish might have been caught in 1976, and it might have been 20 years old. And so we know that fish was born in 1956. Some of those fish got caught in 2009, and they were 5 years old. So we know that it was born in the 2000s. And again, we can look at the banding on those otoliths, and we can look at the size of the bands, because the size of the bands tells us what the growth rates are of that species or of that individual. And these are what the data suggests in terms of growth rates of red snapper over the last like 50 or 60 years. That's a doubling of first-year growth. Like, I always have a hard time believing these data because it's just like the fish are growing twice as fast. And why might that be? I mean, there are lots of reasons. Just three that I put up here is, number one is, I've already said it's getting warmer. And we tend to think of warmer waters being better growth environments. In the Gulf, they have thrown all sorts of structure in the water, oil rigs, artificial reefs. And so snapper might be able to distribute themselves in a way now that allows them to have less competition. Maybe they grow better. Or this compensation idea again. You know, it used to be there were 10 red snappers in the Gulf, and then we killed 5 of them and ate them. These other 5 can now spread out, and so they grow better. And this is one of those compensatory processes. So we accidentally stumbled into something that I think is some pretty cool ecology through these tournament records as well. So for the reef associated, estuarines, the offshore fishes, we see pretty good stories. Like, we don't see anything that's alarming. And then we come to coastal-pelagics. And I'm just going to show sharks here. So what you have now is you have the 3 tournaments broken out, the different colors. We have time again. Sharks were getting bigger and bigger and bigger, probably gears, probably bigger boats. And then you get to like the mid-80s and the bottom just falls out. Maybe in Texas it's happening a little later. But here in Alabama, they're now catching sharks that are like 1/3 to 1/4 the size of what they were catching like 30 or 40 years ago. And this last panel down here is the commercial harvest of sharks in the northern Gulf of Mexico. So in the mid-70s, NIMS, our National Marine Fisheries Service, decided that we could commercialize shark fishing in the Gulf. They even promoted it as a resource. And you see that the catch rates kind of went up rapidly, and then just quickly the bottom dropped out. And so, you know, it's not that these recreational guys, in this case, I'm not trying to make recreational guys saints, but it's not that the recreational guys may be driving this. They just recorded it. If you ask me in this case, it might have been this longline fishery that came in and really drove that pattern. From 1955, when they first started IDing sharks, until 1980 something, the winning shark was a tiger shark or a bull shark every year. Every year, tiger shark or bull shark. And then after that, it's like rarely a bull shark, never a tiger shark. It's like duskies, black tips, hammerheads. So I do think you're seeing a turning over of sharks. Oh, more slides. Again, another table is terrible, I'm sorry, but it's pretty simple. We have our 4 groups, the offshore, the reef associated, this coastal-pelagic group, and then the estuarine group. We have the 3 tournaments. All you really need to focus on is where you see these black dots, because those are the cases where we've seen some increase, or at the end we've seen some decline. Like we've seen some falling off, an increase followed by a decline, or just a decline in general. These are the ones where we seem to have lost the big fishes. And with one exception, it's all concentrated in this coastal-pelagic group, all right? Like there's something about this group in particular that's made them more susceptible to this dynamic. And we are losing the big ones within this group. So what's going on here? Just a few sort of general trends. I'm going to talk real fast here at the end, but it turns out that the coastal-pelagics shown here, they tend to get really big. So it makes sense that they're the ones that you might see the losing of the biggest of the big, because they have that sort of room for change. But interestingly enough, the species where we see losing the big ones, they're actually not species that live very long, as shown here. It's not like the long-lived fishes where we're seeing this decline. It is fishes that tend to spawn kind of fairly late in life. They become mature later, and in fact, that's like the coastal-pelagics. Most of them become mature late in life. And then this last one was just interesting to me. This is the vulnerability score based on IUCN measures for these different species. And the species where we're seeing this loss of the big fish happen to be the ones where the IUCN scores would say they're the least vulnerable. And so there's some mismatch perhaps in what's going on here that I don't actually understand, but I think it's probably interesting. I wanted to close by talking a bit about shifting baselines, because you hear it a lot, and I kind of let it just kind of blow by me sometimes because you hear it a lot. But this study does bring it into specific relief for me. For instance, here's a picture from 2003. Here's a tiger shark. You see the bars. You see this kid here. He's like in awe. Like this is a big fish. Like this fish would scare me out of the water. It's my size, right? And so you like think like, man, that's impressive. And what you may forget is that like just like 25 years ago, they were pulling fish out of the Gulf of Mexico that were 4 times that size. This is a 900-pound tiger shark, right? This tiger shark could literally bite that tiger shark in half. And so you think about the role that these organisms are playing for, like, you know, eating turtles or eating bigger things, like they're probably pretty different. And we don't know how the system has responded. But, you know, today we'd be impressed by this, and you probably should be. But goodness, like imagine when that was a lot more common. To wrap it all up, I talked at the beginning about press and pulse. And this is my conclusion. In the Northern Gulf, I think the press disturbances are probably where the story's at. So what I've shown here is another paper that I was a part of, a fairly minor part, still a part of, where we looked at shoreline armoring. The more reddish colors are the hotter areas. That's where there's more shoreline armoring. But people are armoring these shorelines all the time, and it's just a creep. Like it's just a slow creep, like nothing dramatic, but it's happening. And now we have whole counties where like over half of the shoreline is armored. Like we don't have just a natural shoreline. This is like a press. The warming is a press. The fishing is a press. And the data that I've collected tell me like, hey, that has an effect. I'm seeing it. And then, at least in the case of one pulse that's famous, the oil spill, it got a lot of headlines. And I'm not saying it's not important, it clearly is, but for these coastal fishes, they seem to have been resilient and kind of breeze right through, which I think is interesting. So sign me up as a press man. All right, this is a natural last slide for someone who talks about fish. These are the mangroves. And the case I like to make, maybe for like some of the students is, you might, you know, think of yourself as fairly narrow. Like if you just want to study black mangroves. And what the Gulf has taught me is, man, you can be like all things to all people if you want to be, because these systems are dynamic. The mangroves are creeping up the coast in response to climate. And we don't know what the effects of that are going to be, or the interactions between the mangroves and the marsh. We don't fully understand, especially from the services. It turns out that mangroves, if you look at lab studies, are a lot more sensitive to oiling than marsh is. And we don't know how the mangroves respond to the oil. I've not seen that paper yet. And then we think of this as some sort of habitat for crabs and fishes, but we're not really sure if it's good habitat. And so you can be doing all those things if, just focus on this one plant, but man, it allows you so much scope to do cool things. And I really like that about these systems in the Gulf. And here too, I mean, it's not any different here really. But that just makes me like happy to know that I can go focus on something and drill into it and also sample lots of different issues. All right, I know I ran long, I apologize. I do want to thank sort of co-authors and people in the lab. I get to thank libraries and newspapers, not many people do that. Had some good funding that I have to acknowledge, and then thank y'all for listening. I realize I'm like 10 minutes.
Let's take a couple of questions. Scott, sure.
Yeah. So is the Gulf for some reason really close to, I mean, the changes that you talked about at the beginning are so dramatic. Does that happen often? Like, does it add just kind of like, I know the word's overused, but tipping point that you can get these really kind of strong shifts that you might not see up in more
Right. Well, what does bisect the Gulf is like, a distinct biogeographic break between sort of subtropical fauna and tropical fauna. And that I think does allow you more scope to see those responses. But I wouldn't call it unique in that, I may not do your question justice, but you can probably play at least 2 of the 3 games I played here in North Carolina. So right here at Cape Hatteras, we've got a biogeographic break, and we're already interested in what's going to jump across that. So in the Gulf of Mexico, 50 to 60% of all the fishes that I would sample are pinfish. Here in North Carolina, it's 80%. I mean, so we have a lower diversity fauna. And then in the Chesapeake Bay, pinfish are absent. So like imagine what might happen to the Chesapeake Bay when pinfish make that jump, if they do make that jump. And so like I think there are lots of places that have those setups where you can see some sort of tipping point or something. Like I said, I mean, in North Carolina, I very rarely saw a stone crab when I was a teenager, and now they're everywhere. Come down the coast and enjoy all the ibises, because there are ibises everywhere. And they're nice to look at, and they weren't here when I was, a kid. And so a lot of those things are happening. The fishing effects, the trophic cascade example of sharks and rays and scallops, I mean, it's here. So I don't think the Gulf is like unique, but it is a nice study system. The Gulf is also just really dynamic in terms of the oil industry, and shipping, and fishing. It's such a rich place to get involved in lots of applied issues. That part's probably kind of cool. I don't know, did I help?
Yeah, no, that is. And I guess one thing I would just tag on to is like, because the mangroves are so dramatic on the shoreline.
Right.
Do you have people, I mean, is that something, is there a historic,
Okay, right. Well, here in North Carolina, we're losing eelgrass, probably. Because we're at the southern end of where eelgrass occurs, and we're gaining shoalgrass. And so, like, those mangroves are a nice system, but, mangrove marsh, eelgrass, shoalgrass, I can play those same games here. So, we have a lot to do here. Yeah.
Now, we tend to think of the past as the good old days. But, I mean, if we think about the 1950s and 60s, and I mean, before the passage of the Clean Water Act, the Clean Air Act, I mean, environmentally, we were not better off than we are now. So I wonder if, you may not have seen stone crabs when you were a kid because the environment was worse off than it is right now.
Right.
So is there a way to kind of tease out these effects?
So I think I have something for you. I mean, you make a very good point. And to try to answer that, I would start with is in the very western tip of Alabama that touches the ocean, the town's called Orange Beach. And the reason it's called Orange Beach is because in the 1950s, guess what they grew? Oranges. And they don't grow oranges anymore because the 1970s were a cool period. So the 50s were warmer than the 70s, and now it's gotten warmer yet again. And so I would not presume that this is the first time, for instance, that snapper have shown up in where I'm standing. Probably in the 50s, they retreated a bit. Sure, layered on top of that, there's like fishing effects that I totally would buy into. But we do need to think about these climate signals in terms of the, you know, the cycle, the mini cycles that are on this bigger trajectory we're on. The shark example comes to mind. We're at a place right now where NOAA, if they have a message, is probably that great sharks are recovering. They're surveying off the North Carolina coast and up the coast, and their most recent reports are we're seeing the bigger sharks come back. And so, like, the data that I have here are just, you know, they're where we're at now. They're not like the future for forever. And so I think, yeah, maybe we are going to see recovery of the big sharks because we're not killing them. So sure, I totally agree with you. And I have some examples to try to convince you I'm a believer.
Any questions from IMS?